So in the yesterday class, so I started with the hemoglobin estimation. So I have hemoglobin estimation and the procedure for the hemoglobin estimation, where I told you what is the importance of hemoglobin, why we need to estimate the hemoglobin, and what is the importance of other hemoglobin estimation methods. Like we have also discussed like what are the different derivatives of the hemoglobin, right? And what is its function or purpose to estimate, and what it shows, and the disease condition and all can also be determined by using the hemoglobin estimation. And we were also discussed about what are the normal values in the human beings at different stages, like man, woman, cord blood, and the children at 1 year, children at 10 to 12 years, and so on. Then later we saw various methods of hemoglobin estimation. So where we can estimate and what are those methods. And in these methods, we saw like color-based method, physical method, chemical method, and the astrometric method, and then spectrophotometric method. But out of it, you can see there are some methods where we can see like a visual comparison. Okay, so what happened? So there are so many methods to identify, estimate the uh, hemoglobin, and out of it, the basic method which usually even the ancient people they follow to see to estimate or to come to a conclusion that what is the amount of hemoglobin present in this, or like they can say that by seeing the eyes, or you can say the eyelid, by opening the lower eyelid, and they can see if there is a red uh, color of this arteries. If they saw, then you can automatically they will say that the person is having sufficient amount of hemoglobin. And similarly, you can also see that the people who are you can also see by just looking at the tip of the nail, or sorry, tip of the nail, or you can also like a finger. So by pressing it, you can see uh, the amount of blood present in the sample, or like in the person, right? Or you can say like hemoglobin present in the person. So in that case, a visual method is the one which we can see. Then Talquest scale method is another one, and Dare's method. So these are all like not an accurate method. So they are the appropriate methods which may not be completely followed. Then the WHO hemoglobin color scale is one of the method which can be easily followed for even a rural people can do it. Okay, so we may have to get this, and then we can have it. We can measure the hemoglobin, right? This one. So for that you need only a drop of blood, right? So you are placing that in a filter paper, and then you are checking the color of the sample, right? That's what we are going to see. So WHO hemoglobin color scale is one of the important ones. So where we can use even in the laboratory, or even at home we can use it. So only thing we need is maybe you can download this WHO color scale in your what I can say in your mobile, and then you can do the hemoglobin test at your home itself. So this is a task for you I'm giving. Okay, so you have to be sure that you are doing it, right? Clear? అర్థమైందమ్మా అందరికి?
Yes, sir.
Is everyone clear with this WHO hemoglobin color scale?
Yes, sir.
So you have to take a small paper, filter paper. Then filter paper మీద just ఒక needle తో కుచ్చి ఆ blood drop దాని మీద వేసి, and then color check చేయండి, right? So if you check the color, then automatically what happens, you can see what is the amount of hemoglobin, range of hemoglobin which you have, that you have to report. Okay, na? This is a simple method where we can use and to estimate the hemoglobin. Okay, na? Very simple method. Okay.
Okay, sir.
Okay, so you have to show me this results by Monday. Okay, positively you have to show that in the and by Monday. Okay, so next is Spencer's method. So the Spencer's method is another method where we can see It is also a kind of like, manual method and the color dilution method of the blood. So the oxyhemoglobin will get diluted and matched visually, but it is not appropriate method to estimate the hemoglobin. And when we compare this method with the, because we also see the dilution method in uh, Sahli's method. Sahli's method is the one where we estimate the blood. So whatever the no hemoglobin test we are doing during the in medical camps and all is with the Sahli's method. Okay, so there we are using a small kit, and then that kit will help us to estimate the hemoglobin, right? So we will take the acid and then we'll get the brown color. So the human eye is having an error that when it exposed to the blood color, it may not be identify the blood color. Do you understand what I mean? So it may not be identify the blood color in the sense, so the red color, it may not be clearly distinguished by the human eye. Okay. So కొంచెం little change or కొంచెం change ఉన్నా కానీ, so we feel that the same color. But whereas a brown color is the one where we can clearly identify, clearly distinguish, demarcate the amount of hematin which is present in, like acid hematin which is present in the sample in case of like Sahli's method. Whereas in Spencer method, you can see that the oxyhemoglobin is there, and this oxyhemoglobin, it looks like red in color. So the red in color is not clearly distinguished by the human eye. So that's why we may not be get the accurate value with the Spencer's method, but we can go for the Sahli's method. See what will happen in the Sahli's method then. So in Sahli's method, we use acid. So we already know how to prepare the acid, right? So we use like 1 by n by 10 HCl. So that is 0.1 n HCl, is it not? So we take 0.1 n HCl for diluting the hemoglobin, or you can say that the red blood cells. Is it not blood? Do you remember? Is anyone remember? What? You don't remember? See,
Remember,
Sahlis.
sir.
Yeah, Sahli's method. So whatever the hemoglobin estimations we have done at the NSS camps or in the blood donation camps or even in any medical camp and all. So because we are involved in all those, right? There what we have done. So we did the Sahli's method. Okay. So in the Sahli's method, what are we doing? So we are just collecting the blood, 0.5 ml of blood, and that is diluted with the zero point 0.1 and HCl, right? And then it is keeping in the Sahli's instrument, so where we can compare the color of the blood. Is it not? Right? We will dilute it, and then dilution will give you what is the amount of hemoglobin present in the, on the scale will be there on the each tube, and that scale will represent the amount of hemoglobin present in the sample. Is it? That's what we know. So usually what happens here is the principle involved in this method is like hemoglobin is converted to acid hematin and by 1 n HCl. So n by 10 HCl is nothing but 0.1 n HCl. And the resulting brown color is compared with the standard brown glass reference block, right? So we'll have a reference block in this, and reference block will be compared with this sample. And if it is still dark than the standard, and then automatically what happens? We'll dilute again. So we are going to add some more. We'll dilute again. So once we dilute it, then automatically what will happen? So when we dilute it, so automatically we can also see that there is a, so finally we'll get the dilution, and dilution leads to will give you a reading, or that reading is supposed to be similar to the amount which is present in the, or the amount of hemoglobin present in the sample, right? That's what we will get. Then you can also see the intensity of the brown color is always depends on the amount of acid hematin produced. Right, so the intensity is always directly brown color is always depends on the amount of hematin, acid hematin produced, and which in turn depends on the amount of hemoglobin in the blood sample. So that means 1 molecule of hemoglobin is equivalent to the 1 molecule of acid hematin. So that's why what is the amount of whatever the amount of hemoglobin which we are going to calculate should be there on the reading scale, and that reading scale will help us to identify the hemoglobin levels in a particular person. So the person who is donated the blood or who is going to subjected for hemoglobin estimation, right? And here you can see what is the instrument. Instrument you see like this is a pipette which can be useful to suck the blood, right? So the blood will be placed in the tube. So that means this is a normal tube, and this glass tube will be placed with the blood and then will be diluted with the 0.1 N HCl. But you have a reference block here. You see, this is the reference one. So the reference block always you will see the color, right? So when it matches to this color, you see this color, and this color represents what happens the level of hemoglobin, and these tubes are having the scale. We already know this, right? I don't need to explain you more. Is it not?
Yes, sir.
So all of you know this, right? So Sahil's hemoglobinometer, and it is having like the main parts of which are a graduated glass tube, color, comparator. So you can compare the color with this standard one, and then glass stirrer and Sahlis pipette to measure 20 microliters of blood. So we'll just collect the 20 microliters of blood from here, right? From this, we'll suck that, and then so we'll use that for the estimation. And the tubes commonly used are square with gradations. Gradations in the sense there is a scale on it in percent on one side and grams per 100 ml on the other side. So you can calculate either in percentage or in the gram per 100 ml deciliter. So the how much of hemoglobin is present in the sample. So we'll have like two scales. One side it is having a percent scale, and the other side it is having a gram per 100 milliliters scale is also there. So mostly we go for like grams per 100 milliliter because which is directly can help us to interpret the results, whether the person is having a high amount of hemoglobin or sufficient amount of hemoglobin or insufficient amount of hemoglobin, that we can calculate. Then the color comparators are made of brown colored glass, and same better instruments have a glass prisms. So the glass prism will give you the standard color instead of like glass comparators. Comparators can be like glasses. So uh, whatever the instruments which we have in our laboratory, they are having only comparators, not the prisms, glass prisms. Okay, so we will compare them. We'll compare with them, and then we will see what is the amount of hemoglobin present in the sample. So that's what we can see. Then, so how do you prepare the N by 10 HCl? So the N by 10 HCl always prepared. Like what reagents is required for the what we can say like hemoglobin estimation. So we need 0.1 NHCl hydrochloric acid, and then distilled water. Distilled water. So the distilled water is for dilution. So we need for dilution, and then blood anticoagulated with EDTA. So you should add EDTA to this, and then so that it is anticoagulated. EDTA is an anticoagulator, so that's why the blood can be appear. But if you do it immediately, you don't need to have the EDTA. Because in our case, what happens, we are going to do immediately, right? Instantly we'll pick the blood, and then that blood will be supposed to transferred into the pipette and diluted, right? But whereas if it is in the laboratories, clinical laboratories, they will collect the sample, and the collected sample will be added with the EDTA, which is an anticoagulant, and so that the blood will not get coagulated or like um, what we can say like gaddagattada antam kada, so that will not happen, right? So anticoagulator will be added to it. Then the procedure for this is like, so place 0.1 NHCl in the tube. So n by 10 in the sense it is like 1 by 10. 1 by 10 is like 0.1 N. Okay. అంతే కదా. 1/10 అంటే 0.1 కదా, right?
Sir.
So place n/10 HCl in the tube with the lowest mark. So the lowest mark which appears up to that place, you have to add the 0.1 n HCl. Then draw blood up to 20 millimeters mark in the Shachlis pipette, and then transfer it to the acid tube. So we were already added acid up to the lower mark, and then we are adding the 20 microliters of the 20 microliters, it is 20 microliters mark. So up to that we are taking the 20 microliters of blood, and that blood is added to the HCl added tube, and rinse the pipette well by drawing up and the acid and re-expressing it. Mix the acid and blood by shaking the tube well. So you have to uh, shake the tube well, then mix it well, so that what happens, all the contents will get mixed properly. Then allow it to stand for at least 10 minutes, and to allow brown color to develop due to the formation of acid hematin. So the acid hematin will form when we mix it properly, and when we keep it for like 10 minutes, and then automatically you can see that so it develops the brown color. So the 95% of the hemoglobin is converted at the end of the 10 minutes, right? And only 98% of the color develops at the end of the 20 minutes. So 95% is more than enough to estimate the hemoglobin. So that's why we go for like, we'll keep it only for 10 minutes first on the stand, and then we'll go for the estimation. And if it is in a critical case, and then you have to go for like 20 minutes. Okay. That's what we can see. And then now dilute the solution with the distilled water, drop by drop, with continuous mixing using the glass rod provided. So when we add a glass rod to this, then automatically, so when we mix it properly, so automatically what will happen? You can see that it can be diluted. So the dilution is always something like a dilution should be, it should be comparable with the color which it has, right? So the comparable color should come, right? So that means we have a standard, right? A comparator. So the comparator color should be matched to this tube. Until that, you have to add the distilled water, and then you have to mix it properly. So when it is, when it comes exactly to that color, and then we can say it is having the proper dilution, and that proper dilution will give you like, when you add the distilled water, then it rises up to certain level. So that means in the scale it shows some mark. Up to certain mark it will rise. So that raised point can be counted. You can be calculated as like a percentage, or you can also calculate it as a like a gram per deciliter, right? So the grams of hemoglobin per deciliters, that's what we can identify. And see here, you can see like or I can say like this is one is like comparator block, and then this is the the sample diluted with the distilled water. So now you see if the color is same, so we will place this in this place, and then we will compare the color. If the color is same, and then we can read the reading. So now it is up to 12. So that means, what does it mean? It is 12 grams of hemoglobin present in 100 ml of blood. So the 12 grams per deciliter can be identified for that particular person, right? That we can see. So match the color with that of the glass plates in the comparator, and then while comparing, take care not to leave the glass rod inside the glass tube. So you have to remove the glass rod. And reading is taken when the color of the solution in the tube exactly matches the comparator, and matching should be done at eye level against natural light. Eye level against natural light. So you have to be very sure that so it is having the completely matchable with the naked eye, and it's not with the light. So natural light in this, you can see that there is a disturbance, right? That's why I have to keep that for the dark. then the level of the fluid at its lower meniscus is noted, and the reading on the scale corresponding to this level is read as gram per deciliter. So lower meniscus ante, like see, when we place the liquid, you can see that it can be formed like this, is it not? Like a half moon shape will come at the top of the liquid, right? So that you can take only the lower line as a hemoglobin level, right? So that level will indicate what is the amount of hemoglobin, and that should be represented as gram per deciliter. So this is what we can calculate the uh, hemoglobin level. And what are the different technical errors will happen during the uh, estimation of hemoglobin? So the technical errors which happens are like improper mixing of blood. If you are not properly mixed the blood while estimating the hemoglobin, and then it doesn't help you. And similarly, the errors in pipetting. So if you are not properly pipetted, like Shahil's pipette is there, Shahil's pipette can help you to take 20 microliters of blood. If you take more blood or if you take less blood, and then automatically you are not going to get the exact value of that particular person hemoglobin, right? So that's why the error will happen with the pipetting. So you have to be very sure that you are extracting or you are taking only 20 microliters of the blood. And similarly, once we add the blood and then HCl and then water, you have to be properly mixed. So you have to be very sure that you have to mix properly. If you don't mix properly, then automatically what happens, the brown color may not be sufficient, or like it's not properly distributed among the solution. So when it is not distributed throughout the solution, then what happens, we may get the wrong results, right? So these are the three and you can also see like a tissue fluid contaminating capillary blood. Tissue fluid contaminating capillary blood. So sometimes what happens, from tissues, the blood will be collected, and then this tissue can also be interfered in the hemoglobin estimation. So when the tissue is interfered, then automatically you can't say that it is because of like the hemoglobin content. So you may be having like a tissue may interfere in the acid when we add the acid, and then that tissue will have the interference, and you may not get the exact value of hemoglobin for that particular person, right? And you can also see some visual errors. So while comparing, I already told you that you should not compare the color by keeping it for light. So you should not keep it for light. So it has to be to the dark. So then only you can get like good amount of hemoglobin present in the person, or whether he is an anemic or whether he is having any kind of condition, that we can say, right? So that is how you have to see visual errors, is like taking the reading is very subjective, as it is a comparison of colors. It can be very vary from person to person. So that means one individual calculation and the other individual calculation will be different. If you go for like getting value from the machine, then it's not an issue. So we'll get a value. But whereas when we are comparing the colors, and then automatically you can see that the comparison will vary from one person to the other person, because this is I what we call it as like eye variations can be seen, vision variations can be seen. So the results may not be accurate. This is why the results are not accurate. And quality of the color comparators can affect the reading. So if the quality is not so good, comparator quality is not so good, then definitely it affects the results reading, right? If the glass blocks are old or faded, it can cause wrong results. If the glass blocks are old or faded, it can cause wrong results. What does it mean? So that means if the glass is not clear, so some dirty things are there, or it's not so clear, and it is a not having the readings and all, it is a old one. So when it is old one, so you can see that the so this markings may be erased. So when the markings are not there, then automatically you may not be clearly properly identify the amount of hemoglobin present in the sample, right? That is also possible. So insufficient time allowed for the convenience. So that means the drawbacks are also like errors also. Insufficient time allowed for the conversion of hemoglobin to acid hematine. So that means I already told you like 10 minutes is sufficient to convert the like 95% of the hemoglobin into acid hematine. And 20 minutes is required for the 98% conversion of the hemoglobin into acid hematine. So most of the time, a minimum of 10 minutes is required. So for the reaction to be almost complete so that it will be get done. So otherwise, a biological false negative results is obtained. Okay, so you have to be very sure that you are keeping the sample for 10 minutes. Do you understand this? I think you are not doing it, right? Is it not?
Yes, sir.
So we have to keep it for 10 minutes, right? So remember, so while doing the experiment in the lab, so when you come back to the offline classes, so you have to be very sure that you are doing this. Okay, so you have to keep it for 10 minutes, right? The next is carboxyhemoglobin. So carboxyhemoglobin can form or methyl hemoglobin, methemoglobin, or sulfhemoglobin are not converted to acid hemoglobin, acid hematine. So the carboxyhemoglobin cannot be converted, and methemoglobin and sulfhemoglobin, these are all the three types of hemoglobins which are present inside our body, is it not? Right? So
Yes, sir.
if you get the blood, if you draw the blood, so what happens? We are going to add this 20 microliters. The 20 microliters is also having like normal red blood cells, and it is also having normal hemoglobin and carboxyhemoglobin or methoxyhemoglobin or like sulfa hemoglobin are all there. But these hemoglobin molecules may not be interfere or may not be converted into acid hematine. So when it is not converted into acid hematine, then you can't characterize or like include the hemoglobin value with this three molecules, right? So the hemoglobin value which is coming or which we are calculating is only for the normal hemoglobin molecule, not for the any combinational hemoglobin molecules, right? So non-hemoglobin substances such as like protein, liquid, and cell stroma interferes in the color of the blood, which is diluted with the acid, so which gives the false result. So non-hemoglobin substances which are present in the blood, like proteins, liquids, and cell stroma and all, can also interfere. So that's why you have to be very sure that uh, you are not like drawing all the cells and other things into the blood. Okay, then the time delay. If the brown color of acid hematine is not stable, so undue delay in reading the test results is not allowed. So the test results is not allowed. So when it is not a stable one, acid hematine, if it is not stable, and definitely we won't go for estimation of hemoglobin by using that method. So that means there is something wrong in the method while doing it, while performing it. So that's why you have to go for again, you have to test it. Okay, so the hemoglobin will be tested again. So there is another method which can also be used to estimate the hemoglobin, like alkaline hematine method. So this alkaline hematine method, hemoglobin is converted into alkali hematine. So by the addition of like 0.1 N sodium hydroxide, you see in the previous case in the Sahles method, you see that 0.1 N sodium hydrochloric acid is used. But whereas in the alkaline hematine method, you can see that we are going to add 0.1 N sodium hydroxide, right? And the alkali hematine gives a brown color that can be read against the comparator standards or in a colorimeter. You can also read it in the colorimeter, right? So whatever the color, brown color which is raised, that should be that should be measured in the colorimeter. So what kind of operators we require for this alkaline hematine method? So photoelectric meter with a green filter. So we need a photoelectric meter with a green filter. Then 0.1 N sodium hydroxide, right? 0.1 and sodium hydroxide. So still people are joining, right? Toby. Okay, so 0.1 and sodium hydroxide we need, and then 0.05 ml of 0.05 ml pipette is required, and standard Gibson's or like Harrison's standard is required. Gibson's or and Harrison's standard is required. So this is a mixture of like a chromium potassium sulfate and cobaltous sulfate and potassium dichromate in aqueous solution that will be available commercially in the from the companies. And the solution is equal in color to the 1 in 100 dilutions of blood containing 16 hemoglobins per deciliter. So that means 16 hemoglobin, uh, 16 grams of hemoglobin per deciliter. That's what we can say. And this technique can be done like you have to add 0.05 ml of blood. So that 0.05 ml of blood, that means it is 50 microliters, right? You have to add 50 microliters blood. Then it should be to the 4.95 ml of 0.1 N sodium hydroxide. See, the sodium is always like N and small a, it's not a capital A. Okay. Sodium hydroxide is always capital N, small a, and OH. So it is written here, it is wrong. Okay. And it should be mixed well and boil for 4 minutes. So once we add the 50 microliters of blood to the 4.95 ml, so that means the total volume should be 5 ml, right? So the 4 minutes you have to boil this solution, mixed solution. Then along with the 5 ml standard solution, you should also add the 5 ml of standard solution. So the standard solution will be added and then it is boiled, mixed well, boiled and for 4 minutes. Then cool quickly in cold water and match the test against standard using colorimeter using green filter. So if the test given to high value, like it is added like 5 ml of water and read again. So that means when colorimeter will give only up to 3 OD, right? So when it is more than 3 OD and then you have to dilute it. Like use 5 ml of distilled water and that distilled water will be added to the solution. Like this color, like a quick color will come, right? So that color will be get diluted and then we have to go for the reading. If it is not done again, so again you have to go for dilution, right? And for calculating it, if the OD of the test is like 21 and OD of the standard is like 28, then as the standard is equivalent to 16 grams per 100 ml and the hemoglobin of the test will be like 21 by 28 into 16 grams per 100 ml, that is equal to 12 grams per 100 ml. Okay, so this is how we can calculate the hemoglobin of a particular person. And again, 16 grams per 100 ml, that is equal to 100%. And 12 grams per 100 ml is like 12 by 16 into 100, that is equal to 82%. So it's not like completely you have 100% of the hemoglobin, you have like 82% of the hemoglobin. So the normal person, usually the men will have like 12 to 18 grams of 17.5 grams of what we can call it as like a hemoglobin, gram hemoglobin per deciliter. So 82% is so good. It's okay. It's a normal range. And the advantages with this alkaline method is like it's unlike Shahli's method. Carboxyhemoglobin and sulfur hemoglobin are converted to alkali hemoglobin. So that's why it is most important that here you can see that the carboxyhemoglobin and self-hemoglobin are converted into alkali hemoglobin. So you are going to get the accurate hemoglobin which is present in the blood. Okay, so that's what you can see. Then fetal hemoglobin is resistant to the denaturation by alkali, and this method is used to determine the level of fetal hemoglobin in blood. So alkali method is not sufficient. So that means fetal hemoglobin can be measured by using this alkali method. Okay, so then the disadvantages with this method is like the solution of the hemoglobin in alkali has to be heated to ensure complete denaturation. So the complete denaturation should be done. You have to heat it properly. Otherwise, what happens? The complete denaturation if not happens, and then you may not get the exact hemoglobin value. So that's why it is most important that you have to be completely denaturate. So by heating. So the heating process is the most important process in this case. Okay, in this method. And matching should be done within 30 minutes after boiling. So if not, and then you may be seeing that there is a fading of the color. So that's why that leads to the what will happen. So we may not get the exact value for this. That means a fading always give you the wrong results, right? That we can see. Okay, so is it clear for all of you? Right?
Yes, sir.
Yes,
The
sir.
two methods what we discussed today are like Shahli's method. Shahli's method is the one which we follow in our laboratory, right? So the Shahli's method is a general method which we are following, right? And besides that, we were also discussed about the other method like alkaline hematin method, right? So the Shahli's method is having like acid hematin, and whereas in alkaline hematin we use sodium hydroxide, and whereas in case of Shahli's method we use uh, hydrochloric acid, right? But the Sahli's method is the most feasible method that we can do anywhere. But whereas in case of like alkaline method, alkaline hematin method, so you have to go for like heating and all, right? Or you have to dilute it and then see the color and so on. So that's a little bit difficult method when compared to the Shahli's method, right? And there is another method called as acid alkali method, right? So this acid alkali method is like this is a hematin method, alkaline hematin method. The solution of hemoglobin has to be heated to ensure complete denaturation. So in the previous one, right? And this procedure can be omitted if the blood is collected first into the acid and then transferred, and after standing for 20 to 30 minutes, sufficient alkali is added to the neutralized acid and convert the acid hematin to alkaline hematin. You see, this is a mixed method where we can see like it's a combination of like Sahli's method and alkaline hematin method. So in the alkaline hematin method, what happens? We have to go for like heating of the sample. Denaturation has to be done. But whereas in acid alkali method, what we are doing is we are going to take this. We are going to omit this method. I mean this heating step. Then the blood will be collected, and the collected blood will be immediately placed in the acid tube. Immediately placed in the acid tube. So then that will be collected like that is what we are doing in our experiments, right? When you are doing the hemoglobin estimation, so we are going for immediately we are placing the blood sample into the acid, is it not? That's what we do. And this is standard for 20 to 30 minutes. You have to stand it for the 20 to 30 minutes, and sufficient alkali is added. So we will also add the sufficient amount of alkali to neutralize the acid, so that what happens, the acid hematin will be get converted into alkaline hematin. So that alkaline hematin can be measured. Then procedure for this is like add 0.05 ml of blood, 4.95 ml of 0.1 HCl. The same is like alkaline hematin method. And after standing for 20 to 30 minutes, add 0.95 ml of 1N sodium hydroxide, and the tube is inverted several times. That means mix it properly, right? And after standing for not less than 2 minutes, the test sample can be matched in photoelectric colorimeter using the yellow-green filter using Gibson and Harrison standards. So because of that, what happens, we can calculate what is the amount of hemoglobin present in that. Then Haldane carboxyhemoglobin method is also there. That means we can calculate carboxyhemoglobin. But in case of a alkali hematine method, you can see the other two types except the carboxyhemoglobin. Other two types of hemoglobins can be measured, right? So they can also be included. But for carboxyhemoglobin estimation, you have to go for the Haldane method. So in the Haldane method, hemoglobin is converted to carboxyhemoglobin by the action of coal gas on the diluted blood. So apparatus required here is like Alden's graduated tube and standard, approximately 0.4% of ammonia in distilled water, and then 0.02 ml of pipette. 0.02 ml pipette is required. That means 20 microliters pipette is required. And fill the graduated tube up to the 20 mark with ammoniated distilled water. Then add 0.02 ml of the patient's blood and mix well. And pass coal gas through this solution for 2 to 3 minutes by means of rubber tubing attaching the pasture pipette, so that we can have the gas supply, and dip the end of the pipette into the capillary alcohol and gently double the gas through the blood solution. So the capillaries, the caprylic acid prevents the frothing, so that add 0.4% of ammonia drop by drop you can add, then mix them between each addition. So until the solution view the diffuse daylight, viewed in the diffuse daylight, and match the standard. So you can match the standard so that you can know what is the amount of uh, sample present in that. So read the amount of solution in the calibrated tube, and the calibrations gives the hemoglobin concentration as per the percentage. And calculate the amount of hemoglobin in the gram per 100 ml of blood. And for example, if the color standard is defined as corresponding to 14.6 grams per 100 ml and reading is like 95, then the hemoglobin content is calculated as like 100% is equal to 14.6 grams per 100 ml. Okay, so that's what you can see here, that 14.6 grams and then per 100 ml, and 95% అయితే, so you have to calculate like if it is 100%, it's okay. If it is 95%, then you have to go for like 14.6 into 95 by 100. That is equal to 13.87 grams of hemoglobin per 100 ml blood. Okay, so this is how we can calculate. And portable hemoglobinometers are available. So using this portable hemoglobinometer, so we can calculate the amount of hemoglobin present in the blood sample. Do you have the class now at 3 o'clock?
Yes, sir. Yes, sir.
Okay. Okay, so I have like 5 more minutes. I'll tell you what is spectrophotometric method, right? So we can use like you can cyanomethoxy uh, sign meth hemoglobin, met hemoglobin, or like oxyhemoglobin can be measured by using the spectroscopic methods or spectrophotometric methods by measuring the density, optical density, right? Oxyhemoglobin can be measured, so using different protocols. But you see, I'm not going to tell you that all procedures which are related to this, but remember, we have like two methods. One is called as alkali hematine method, or you can also see like a Shahli's method, right? Shahli's method, which we are following in our laboratory, and we can also follow the other method like WHO color comparison method, right? I already gave you the exercise, right? You have to be very sure that you are going to you are going to test your blood and hemoglobin, and then kindly tell me the results on Monday. Okay. So you have to tell me the results by comparing, download the WHO blood strip, right? Hemoglobin color strip, and then compare your blood color with that particular color, and then tell me what is the amount of hemoglobin present in your blood sample. Are you clear?
Yes, sir.
Is it clear for everyone?
Yes, sir.
Okay, so then, so I have to see that hemoglobin should be from your blood sample, right? I want to see that. Okay, so if it is done, and if you have class at 3 o'clock, and then I'm going to take your attendance. So I'll download the attendance. So the 2 members were not here. They may be not joined in the class or something. So they are not here, maybe. Okay, so one more thing I want to tell you here is, I was already posted a Excel file in our WhatsApp group, right? So that WhatsApp group is having the EDU emails of individuals like me. మీవన్నీ ఉన్నాయి. ఓపెన్ చేశారా, చూసారా? రైట్? చూసారా, చూడలేదా?
చూసాం సార్.
Okay, so then you have to enter into the Teams app and then you have to register because you already registered that. Okay, మీకు ఆల్రెడీ టీమ్స్ యాప్ లో ఇంతకు ముందు yogavemanuniversity.ac.in ఉంది కదా, రైట్? @yogavemanuniversity.ac.in తో ఉంది కదా మీ మెయిల్, రైట్?
Yes, sir.
So make sure that you are not using that anymore, and you have to use this like new mail IDs were sent like @yvu.edu.in. Okay, yvu.edu.in. So you have to be sure that you have to do this. Are you clear, ma? మీరు మళ్ళీ రిజిస్టర్ చేసుకోండి. ఓకే, దానిలోంచి లాగౌట్ అవ్వండి. టీమ్స్ యాప్లో నుంచి లాగౌట్ అయ్యి ఫ్రెష్గా ఈ మెయిల్ ఐడీస్ యూస్ చేసి యూ హ్యావ్ టు లాగిన్. ఓకేనా?
Okay, sir.
Okay, sir.
అందరికి అర్థమైందా? Everybody has to remember this. Okay.